So using Biopython, we can deal with the sequences of, like, for example, we can deal with biological sequences such as DNA, RNA, and protein sequences. So what are biological sequences, you may ask? Biological sequences are fundamental components of living organism, like they represent linear arrangement of building blocks of our body is made up of nucleotides and amino acids. Their sequences encode the genetic information and the instruction necessary for functioning, development, and evolution of all living organisms. So there are two main types of biological sequences: DNA and protein sequences. So how we can deal with this? So using Biopython, we can read and write biological sequence, and we can also calculate our sequence properties, like their mass to charge ratio, like their weight, how much they weigh, and how much amino acids are there, and things like this. So if we move on to our first code section, we can see there is a section reading and writing sequences. So using this, first of all, we are going to import the SeqIO module. And using the SeqIO, we can read our sequence.FASTA, and we can also declare its file format, which is FASTA file format. And after that, for sequence, we can iterate our sequence and print their ID and length. We can print the ID of the sequence, or what is the ID of sequence. So here we can see it is the ID of the sequence, and print how much length this sequence has. For example, if I go to the folder and open sequence FASTA, here I can see here is the ID and here is the length of the sequence. So we can see it consists of 451 nucleotides length. Also, we can calculate our sequence property if we import the module biosecutils and import the GC. We can calculate our sequence. Here is our sequence, and we can calculate our GC content of the sequence. So we can create a variable which is GC content, and then we can literate our sequence over here, and then print how much GC content is present in percentage in our sequence. So here you can see there is 50% GC content which is present in our sequence. Moving on to the sequence alignment section. Sequence alignment, arranging the sequence in bioinformatics. So if you know about alignment or you are familiar with this concept, you may also know the sequences. So sequence alignment is a way of arranging protein or DNA sequences to identify the regions of similarity that may be consensus of evolutionary relationship between the sequences. If we know that adenine is also present in another protein, which another nucleotide, which is also adenine, we can compare these two sequences and we can find the evolutionary relationship between those sequences. So interpretation. If two sequences in an alignment share a common ancestor, mismatches can be interpreted as a point mutation and gaps as endels, that is insertion or deletion mutation, introduced in one or both lineages in the time since they diverge from one another. So there are two types of alignment method. First of all is global alignment and then local alignment. What is global alignment? Calculating a global alignment is a form of global optimization that forces the alignment to span an entire length of the query sequence. For example, global alignment is aligning the whole sequence that is present here. And by contrast, local alignment is just comparing the sequence like this. There is here, if we can just, here you can see comparing this sequence is local alignment section, and if we compare all the sequence is called global alignment. So local alignments are often preferable but can be difficult to calculate. To identify the region of similarity, a variety of computational algorithms have applied to this problem. For example, if we just have to compare this section, this is called local alignment. And then there is multiple sequence alignment section. Multiple sequence alignment is an extension of our pairwise alignment to incorporate more than two sequences at a time. So multiple alignment method try to align all the sequences in our given query set. So if I'll just move to our VS Code, here you can see I have imported the pairwise section, and then we have our first sequence, and then we have the second sequence. Now we are going to align the sequence by using the global alignment. We are going to compare these two sequences and find the mismatches between them. So here is the mismatch. So now you can see that there is a gap alignment in it. So that's for the basic sequence alignment that you can perform using Biopython. Now let's move on to our local alignment section. Here you can see I have imported the pairwise alignment module and then also imported the format alignment. So here I have defined the scoring function, how much score for your match, how much score for your case mismatch, and then your gap opening penalty and your gap extension penalty. So here are all the scores that are given. So I'm going to define the sample sequences. So sequence 1 is ATCGA CTTAGC, and then you can see the sequence 2, there are the variables that are defined here. Now we are going to make a function that will perform the local alignment using all these modules, the sequence 1, sequence 2, match, open, mismatch, score, gap opening, gap extension, or these scores, and then I'm going to print the alignment. So here you can see I am going to use the alignment in alignment section and then format this alignment overall. And then we can move on to the global alignment section. So just like local alignment, we are going to import our section and then also define the our functions. The thing that is changed in global alignment is that in performing the alignment, we have changed the function from local to global multiple alignment. And then here you can see here is a global, here is the local alignment, and then we are going to print the alignment as it was. And here you can see there is the whole thing that we performed in this two alignment section. So using the multiple sequence alignment. So multiple sequence alignment contains some different modules. First we have to import some modules. So we have multiple sequence alignment module, align io.module, Seq module, and SeqRecord module. So then we are going to define our sequences. So sequence record, we have the sequence 1, sequence 2, sequence 3, and then we create a multiple alignment object. Here you can see multiple alignment object is created for the sequences, and then we are going to save the alignment to a alignment file. Here we can see the alignment.fasta is the file that we created, and here you can see sequence 1, sequence 2, and sequence 3 alignment with 3 rows and 20 columns was saved. So here if we go, if there is a file which is called alignment.fasta, here you can see unknown description, and here you can see there are 3 alignments of 3 sequences. So moving on, we should focus on a different topic now. So the topic is motifs. So in bioinformatics, a motif refers to a short and conserved sequence pattern, like a short sequence which is present in the whole sequence of our alignment, which can be DNA, RNA, or protein. The motifs are associated with the specific functions, specific structure, or your regulatory element like data box, G box. And identifying and analyzing these motifs are essential for understanding the biological process, as they can provide insight into your gene regulation, your protein function, your evolutionary relationship, your gene networking, your gene expression profiles, so your cis-acting regulatory elements. So for that, we can also search motifs by using the Python. First, we have to import our module RE. So here you can see the support for regular expression. This module provides regular expression matching operations similar to those which is found in the Perl operating language. And then we are going to import the sequence because we are working with the sequence, and we are going to define a sequence. And the motif that we are going to define is CTG. So if I am correct, we have to first give that motifs. So for that, after that, we are going to create an object which is going to find the motif in it, and the motif matches are three. So there are three CTG sequence that are present in our query sequence. So moving on, we have to focus on transcription and translation now. So if I'm correct, we already discussed transcription and translation in the starting lecture. But transcription is the process of copying genetic information from your DNA to your RNA, and your translation is the process of using the molecular mRNA sequence to synthesize a functional protein. So how we can do that? So first of all, we have to define our DNA sequence, and then we can use that DNA sequence to transcribe, and then use that sequence to translate into our RNA. So here is the code that I have defined here. You can see that DNA sequence is defined into RNA sequence, and RNA sequence is defined into your protein sequence. Now we should move on to molecular weight. So the code that is I'm going to discuss is calculate the molecular weight of given DNA sequence, which represent the total mass of all atoms in a sequence. This can be helpful in your various biological applications, such as understanding the composition of your DNA molecule and designing the primers for your PCR. So we have to import the necessary function and classes from Biopython. After that, we are going to define our DNA sequence using our Seq class. And then we are going to calculate the molecular mass of the DNA sequence using the molecular weight function. And then we are going to print the calculated molecular mass. And here are the results that you can see here. So discussing the BLAST, what basically BLAST and how we can use BLAST to identify the organism. So BLAST is a very powerful tool and widely used tool for organism identification. If you have unknown sequence that you want to identify, you can always use BLAST for that, including your microbiology, environmental science, and forensic analysis. It provides a rapid and effective way to determine the potential identity of the unknown sequence by comparing them against vast collection of known sequences in the databases. However, it is crucial to use BLAST result cautiously and consider additional evidence and experimental data to validate that identification. So we can use the BLAST for the sequence retrieval, for BLAST database selection, and use the BLAST tool to perform a sequence similarity also as well. We can also use the BLAST for the scoring and ranking of each alignment section. And then we can also analyze the BLAST result to determine the best matching organism. And after that, we have to validate our results carefully. So now let's look at the code, how we can do that. So first of all, we are going to import our libraries. And then we are going to set some BLAST parameters, like we are going to use the BLAST program, which is a database for protein. And then we are also going to choose a database. So database is NR, which is nucleotide database. And then we are going to give a query sequence. So I have here this the query sequence. So if I search this query sequence in my web browser, so let's just see, if I open the NCBI gene database and search for this gene or protein. If I'm going to search this in the gene database, let's see if it gives us any known information. So we'll wait, and it is giving us a black stripe live bear
identification. So now let's just perform our blast result using the code written, and then read and praise the blast result, get the description of our top hit that we got, and then extract the information from the description using the regular expressions, and then print the organism information and save the blast to our result file. So here we are seeing that it is giving us this Popilius Porophila organism name. And if I go to my database and search this, you are seeing that it is giving the similar results. So we can use the Biopython blast to identify the organism for its identification. This was a known sequence. We can also use query, we can also use sequence as well. So that's it for the whole lecture of understanding our basic Biopython for sequence analysis and how we can use this to identify organisms as well. How can we align these sequences? And in the next module, we are going to discuss something new, something very exciting. So stay tuned for that one. Thank you so much for your attention.